down again today, so I did my best to draw all this out on the board, but just in case, I'm coming home and making a video to correspond to our slides to help you better understand these viruses and the nucleic acids, just because it's that important. All right. All right, nucleic acids. Primarily, we're talking about DNA and RNA, and these are the essential molecules that store and transmit our genetic information. They're going to direct protein synthesis and our cellular functions. I always liken it to being like the recipe card for making all of our great substances that we need in our cells. DNA and RNA are polymers made out of nucleotide monomers, and each nucleotide contains a sugar. In the case of DNA, it's deoxyribose, and in the case of RNA, it's just ribose. Also a phosphate group and a nitrogenous base. The DNA ends up forming a double helix, storing the genetic blueprint. Whereas in our eukaryotic cells, the RNA is typically single-stranded. It's made from copying one of the strands of the DNA, and it's involved in the expression of the DNA information into proteins as part of transcription that leads to translation. All right, so DNA, primary molecule that encodes genetic blueprint for an organism's traits, is passed from the parents to the offspring, has a double helical structure, and the strands are anti-parallel, meaning one runs in the 3' to 5' direction, whereas the other runs in the 5' to 3' direction. And they're held together by hydrogen bonds between the base pairs. We'll talk about that more in a little bit. Whereas RNA in eukaryotic cells, like I said, will play a crucial role in protein synthesis as messenger RNA, carrying the genetic information from the DNA out to the ribosomes. And there they'll get translated into the proteins. All right, so let's understand what the nucleotide is. The nucleotide is the building block of nucleic acids. Like I said, made out of a 5-carbon sugar. In DNA, it's deoxyribose. In RNA, it's ribose. They're attached to phosphate groups, and this will form a strong repeating sugar, phosphate, sugar, phosphate, sugar, phosphate backbone that we see as the kind of ribbons of the alpha helix. Attached to the sugars will be these nitrogenous bases, adenine, guanine, cytosine, and thymine in DNA, and they're hydrogen bonded. And I always say, think of the hydrogen bond as not a strong bond like a covalent bond, but kind of a loose association. All right. But in sum total, we're going to have a lot of hydrogen bonds holding our DNA together. Now in uracil, in RNA, we replace the thymine with uracil. And if you don't remember all this, there's more videos on the structure of DNA and RNA and nucleic acids. The early chapters of our textbook review this, and I have review material on YouTube. All right, so our central dogma of molecular biology is this concept that within our cells, we contain DNA. And like I said, the DNA is the genetic information. It's how we store in cells encode the genetic blueprint for an organism's traits that's passed on from parent to offspring. It gets transcribed into RNA, and I should almost write a little M here as messenger RNA, which is what carries the information from the DNA out to the ribosomes where it can then get translated into the proteins. And it's always this idea that the information flows from DNA to RNA to protein. We never flow back, say, from protein to RNA. All right, so let's now talk a little bit more about the enzymes that are involved in this because they're very important. DNA polymerase. We call it DNA polymerase because it's going to polymerize or synthesize a new strand of DNA. And when we get very specific within our cells, we talk about a DNA dependent DNA polymerase. DNA dependent DNA polymerase means the ending part, DNA polymerase, that we're going to synthesize a new DNA strand, okay? Putting the words DNA dependent in front of it means that we're going to use DNA as our actual template, all right? So these, when we're using DNA polymerase and say our eukaryotic cells, our human being cells, these are the enzymes that are crucial for DNA replication. And we use DNA replication so that we can repair damaged cells. We can grow and develop and create more cells. We can go through mitosis. In class today, we reviewed the whole process of the cell cycle. And DNA is replicated in interphase, specifically the S phase of interphase. And in interphase, S phase, we use these DNA-dependent DNA polymerases to use DNA as a template and initiate the synthesis of new strands of DNA, which is done in a semi-conservative manner. The DNA-dependent DNA polymerase will read an existing template of DNA. So we start out with our DNA that's wound together as two strands. One strand will run in the 5 direction, one strand will run 5 to 3 direction, one strand runs in the 3 to 5 direction. And we open up that double helix of DNA. And then here down represented in pink, we see a little bit of an RNA primase. An RNA forms a little starting primer, okay? But then DNA that we see here in blue, the DNA polymerase takes over, and off of this template strand of our already existing 3 to 5 direction strand of DNA, we synthesize another strand that corresponds to it in the 5 to 3 direction. If this was an adenine, this would be a thymine. If this was a cytosine, this would be a guanine, etc. All right? Here you can see this a little bit better as the overall picture. We started out with our original 3 to 5 strand of DNA right here that was bonded to our 5 to 3 strand of DNA. These orangish colored strands are our original strands. And because they're hydrogen bonded together, where the base pairs right here, where cytosine pairs with guanine, adenine pairs with thymine, they're held together very loosely. And it's important to understand that those hydrogen bonds are kind of loose bonds in sum total, because as you can see, we have so many of them. All of these are going to be correspondingly hydrogen bonded over to the other strand. There's strength in numbers, but every individual bond of these bases to each other is only a loose association. It's a hydrogen bond. It's not a real bond, okay? It's not like a strong covalent bond. So we can, what's important about that is we can unzip or open up our DNA. And that allows that DNA polymerase to get on here and start synthesizing a new corresponding strand of DNA. Helicase is the enzyme that kind of moves down this zippered together right here, structured together, base paired, hydrogen bonded at the nucleotide bases, adenine to thymine, cytosine to guanine. It's what unzips it and opens it up. Opening it up allows DNA polymerase to hop on this original strand and start synthesizing a whole new strand of corresponding DNA. Similarly, on the opposite strand, DNA polymerase will also synthesize another strand. Important to know, DNA polymerase only synthesizes in the direction 5 to 3. So we see it synthesizing a whole new continuous strand shown in green here that we end up calling our leading strands right on this side because it's synthesized continuously, no interruptions. Whereas when we look at this strand is the strand that's in the 5 to 3 direction. It's like, oh my gosh, our DNA polymerase needs to move from 5 to 3. So we're really going to have to open up this whole helix, and DNA polymerase is going to have to move almost, I guess, backwards, for lack of a better term, down the strand this way because it can only synthesize in this direction. So it's needed helicase to open this up. And when it moves down on this other strand, it has to do so in kind of a patchwork manner where it only does a number of nucleotides at a time, stops, does some more nucleotides, and these are called Okazaki fragments, these short little fragments of DNA. We'll end up having another enzyme that comes in and kind of anneals or stitches these two fragments together, and that will be called, DNA ligase, all right? Not as important for this class, but if you were taking Bio 155, which is from where I took these slides, you would remember that. Okay, so our leading strand goes really fast and efficient, all in one direction. Helicase is opening this helix up ahead of it, and once that helix is open up, we can get DNA polymerase to synthesize or replicate our DNA in short Okazaki fragments that we then have to ligate together. All right, so another slide kind of showing everything. Our DNA dependent DNA polymerase. Once again, this would be like in your eukaryotic cells. This would be like at a time you're growing and developing, you need to create some new cells. Your DNA opens up, you need to replicate. Okay, we are going to make new cells where one strand, we call it semi-conservative because you'll have one of your old strands with the new strand that you're making. Again, you'll have one of the original strands, we call that the parental strand, with the new strand that you're making. This kind of flipped from the previous slides, where here we have the 3 to 5 parental strand, and we have our leading strand being synthesized continuously with our DNA dependent DNA polymerase. Here you have the 5 to 3 original strand, on which we end up having to have helicase open everything up before our DNA polymerase can get on here and kind of make these little Okazaki fragments that'll have to be stitched together. All right, helicase is our primary enzyme that unwinds the alpha helix. Unwind the alpha helix, helicase separates those two hybridized strands. All right, and ahead of it, because as we're unwinding the strands, as we're untwisting in that, you can cause supercoiling or mega twisting of these strands that are ahead of that helicase enzyme. So we have another enzyme called topoisomerase. Topoisomerase is the class of enzymes. DNA gyrase would be getting to a specific name of the enzyme. We can somewhat use them interchangeably here. And what they do is they put like little nicks in the strands here so we can uncoil those supercoils ahead of the helicase. And these enzymes are actually important. We'll see that some of our really, really important antibiotics, like levofloxacin, will act at topoisomerase. So I'm not just telling you this for my own health. Well, maybe for my own health if you're my nurse one day, but I'm trying to drive it into you that learning this is important. You're going to have to learn this in pharmacology. It's good to know in nursing. The topoisomerase will be the enzyme that will be inhibited by specific antibiotics. Okay. And these are called the fluoroquinolones. Like I said, levaquin is an example of that, levofloxacin, and they're crucial for inhibiting topoisomerases in bacteria. And when they interfere with that enzyme function in bacteria, they disrupt that microbe's ability to make its DNA. So you can see that's really important. And fluoroquinolones are one of the most prescribed groups of antibiotics in the country right now. I think it's like 40% of all antibiotic prescriptions are in the fluoroquinolone class. All right. So then in our regular central dogma of molecular biology in our eukaryotic cells, we try to go from DNA to RNA, specifically our messenger RNA, using our DNA dependent RNA polymerase. I like to remind you that in our nucleus, we have our double helix of DNA. It never leaves our cell nucleus, all right? It gets transcribed into messenger RNA. And the messenger RNA is single-stranded, small enough it can get through the nuclear pore and move out to the cytoplasm, where it's going to get in the ribosome. It gets in between the smaller 40S ribosome and the larger 60X portion of our 80S ribosomes and gets translated into proteins there. All right. We specifically, to make mRNA, use RNA polymerases. We're polymerizing or making RNA. But again, we call it DNA Dependent RNA Polymerase because we make this messenger RNA off of a DNA template. All right, just trying to ensure that you understand the naming of these enzymes. Okay, so back to looking at another slide of our central dogma of molecular biology. We have here DNA polymerase. In our case, we talk about DNA dependent DNA polymerase. This is used for making new copies of DNA. This is when our cell is replicating. Our cells are growing. They're dividing in half. We're making more of our cells, all right? So we want to keep ensuring that every new cell that we make has a copy of our DNA. We use our own DNA as our template for that, and then we polymerize new copies. So we call it DNA-dependent DNA polymerase. This is the replication cycle going on in the nucleus. Versus when we want to transcribe this RNA. I'm sorry, it should be moved over here to RNA. I don't know what happened to the slide. Let's actually see if I can get out of here and try and fix that for a second. Go back to that slide. I'm just making more of a mess of it. Sorry. I'll have to fix this later, won't I? We'll put RNA right here. Make it a little bit bigger. All right, and over here will be our protein. Oh, what did I just get rid of? All right, I'll put this here. Hopefully this will make better sense to you if I at least have this in somewhat of a manner that looks there, a little bit cleaned up. All right, so we've gone through DNA replication. I have this yellow outline here reminding you that this is in the nucleus and that transcription starts in the nucleus. In the nucleus, we're using our DNA dependent RNA polymerase. All right, so DDRP, whereas here we would have DDDP, DNA dependent DNA polymerase. So it uses the DNA as a template. We open up that DNA and we're going to make a copy of our RNA. And we make a copy of our RNA in what we call the 5-to-3 direction. We also can refer to that as a plus strand of RNA. And this is actually messenger RNA. This messenger RNA is moved out into the cytoplasm through the nuclear pore, and it starts to move over to associate with our ribosomes so it can get translated into protein. All right. Now, boy, is this slide a mess. I'm telling you, what did I do here? I got this word RNA. I don't know what I did. So I'm sorry about this. All right. So when we have viruses that are double-stranded DNA viruses, they're pretty much going to follow the same flow as our central dogma of molecular biology as what's happening in our cell. All right. They can even come in and hijack, and we'll see how they can use our DNA polymerases then to make their DNA. All right. Where things get tricky is when we have the viruses that have RNA as their genome. They have to use a different mechanism because you can see if RNA is their genome, everything is going to need to be made off of an RNA template. Here we see DNA dependent as our template. Up here, I should have written in DDDP. DNA would be our template. Okay, let's go back. You can even write that in there as DNA dependent. Ooh, I can't type. I've done a lot of editing on this slide, haven't I? All right, so it's DNA dependent DNA polymerase. Again, DNA dependent RNA polymerase. They're all depending on DNA being there as a template. Well, when we have an RNA genome, we're going to need to use RNA as our template. All right, so that's where we're going to see the trickiness comes in. All right, when we look again, just keeping in with our own cells or what we'll see with DNA viruses, we have two strands of DNA. We have one strand right here running in the 3 to 5 direction, all nice and pink looking right here, okay? If you trace it all out, ends up in the 5, all right, 3 to 5. And we call the 3 to 5 direction, we call that the minus direction, or we refer to that as the minus strand. In our normal cells, when the alpha helix is all closed up and annealed together, the 3 to 5 strand is associated with a 5 to 3. I'm sorry, I fast on the trigger here on my computer. The 5 to 3 strand. And I kind of traced over a little green line just so you can follow it through. So it's always going 5 to 3. But remember, we had our helicase come open, and it opens up this region of the DNA so that we can open up our strands to transcribe a gene. And keep this thought too, that in your whole DNA, usually you're not transcribing the entire range of your DNA molecule into a molecule of messenger RNA. You want to transcribe an isolated gene. So you end up opening up that region where the gene is on your DNA, and off of the 3 to 5 strand, you can synthesize readily a 5 to 3, this blue strand of RNA using your RNA polymerase. And hopefully you now know that more so we can call it DNA Dependent RNA Polymerase because it's using this 3 to 5 strand of DNA as its template as it synthesizes the corresponding messenger RNA in the 5 to 3 direction. And we always synthesize our messenger RNA in the 5 to 3 direction. All right, so messenger RNA growth is in the 5 to 3 direction. What does this mean? This means you're always adding the new nucleotides at the 3' end of the growing tip. Okay, it has directionality. Again, here's our DNA, the 3' to 5' strand and the 5' to 3' strand. All right, and we would make our messenger RNA to correspond. If this is A here, we would have U here. If this is T here, it would be A here. And here's another thing important to point out. I always said A corresponds to T, C corresponds to G, but as soon as we get into making our RNA, we substitute uracil as the nucleotide base instead of thymidine. So we have substitution of U for T. All right. Once again, looking at our DNA-dependent RNA polymerase coming in here. Here we have, again, you can see A corresponding with T, C corresponding with G, G corresponding with C. When we have an A, it corresponds to a U instead of a T on the RNA. This mRNA is growing from this 5' direction to the 3' direction right here. All right, I love this slide, and I believe the next one is similar. It shows you how at the very micro level we have all of our wound up DNA. Remember how this was all wind around our histone proteins to stay organized? And now we're kind of blowing it up big so you can see this strand running in the 3' to 5' direction of DNA opened up as this strand runs in the 5' to 3' direction. We call the 3' to 5' direction, like I said, the negative strand. We also can call it the anti-sense strand. We'll explain that in a minute. Whereas the 5' to 3' strand will be called the plus strand, also called the sense strand. When RNA polymerase hops onto our 3' to 5' antisense strand, that's the 3' to 5' antisense serves as a template on which we can build a sort of mirror image or opposite corresponding strand of RNA that's going to grow in the 5' to 3' direction. So we can also call the 3' to 5' strand of DNA the negative strand, the antisense strand, and also the template strand. RNA grows 5' to 3'. So we call this the sense of it, okay? It's the sense that the ribosome is going to read. So A pairs with U, T pairs with A, G pairs with C, A pairs with U. Now, if you look at the sense strand of DNA, it's also running in the 5' to 3' direction. It reads exactly the same letters, but every time you see where a T is, you would have to substitute a U for RNA. But otherwise, AC, here again, you're substituting a T for a U. So you really could, instead of having to figure out the corresponding pairs, you could have just written this whole thing out and just substituted a U everywhere there's a T. That's why they're both called the sense strand. The sense strand, the plus strand, the coding strand. It's the DNA strand that is identical to the messenger RNA strand, except we substitute uracil in the messenger RNA for the thymine. You can see when we get our coding strand of the DNA in the 5' to 3' direction, and here it's paired oppositely with the 3' to 5' DNA strand, we could figure out easily what the mRNA is going to be made off of this. You could do it again one of two ways. You could put exactly the coding strand and just substitute U everywhere there is a T, and you can see that that is the same between these two. Or you could figure it out by doing the corresponding. T pairs with A pairs with U, C pairs with G, C pairs with G, etc. If you learned this in the past, you know that you can look up on our universal code chart. Every 3 codons codes for a specific amino acid. So if you look up AUG, it codes for methionine. GGG, it codes for glycine. CUC codes for leucine, etc. I could give you an exam question, and I used to love to do this, where I gave you, say, just the template strand of DNA, and I wanted you to figure out the coding strand, I wanted you to figure out the mRNA, and you could also look up and figure out the primary sequence of the amino acids that make the protein. And I passed out something in class today where you could do that a little bit. And if you just work on that a little bit, it helps make sense in your brain. Just take the little bit of effort it does to try to figure that out, and you'll be like, oh yeah, okay, now I get it, and it'll make sense to you. All right, here's another slide showing the same thing. Once again, we call it the sense, the coding strand, or the plus strand when it's running in the 5' to 3' direction. The mRNA will correspond to the same order in the DNA, only substituting uracil for thymine. So once again, the antisense is the non-coding strand or the template strand. We also call it the negative strand because it runs in the opposite 3' to 5' direction. But we need that template to build our mRNA off of it. All right. So when we look here at our template strand, AGT, it's going to pair A with U, G with C, T with U. Our next one is going to pair C with G, T with A, C with G, etc. So there you get your messenger RNA running in the 5' to 3' direction. If you looked up all these codons on the universal codon chart, you would see UCA codes for serine, GAG codes for glutamate, GUG codes for valine, etc. All right, now as we do this, we'll see that as we get into looking at our viruses, I always kind of find it ironic that we call it the life cycle of viruses when we all know that viruses aren't living, but we kind of use that terminology, all right? So we're stuck with it. And how they basically get into infect our animal host cells, because we're primarily concerned in this class with how the viruses are going to infect us as humans, since that's who we're going to be treating when we go on to our clinical careers. And we'll see that when we have viruses that have their genome made out of RNA, they're going to have to overcome two big problems, in what we know as to how to transcribe and translate into proteins. The first is that our host cells do not have an RNA polymerase that use RNA as a template. There is no RNA dependent RNA polymerase. Our host cells have in the nucleus a DNA dependent RNA polymerase. Okay, so that's a problem for our viruses that have RNA as their genome. What the heck are they going to hijack to start making copies of their RNA? And then they're going to need to translate their viral RNA into protein. All right, so translation is going to have to begin on RNA, and typically it's going to translate into a single polypeptide. Now, if the RNA is the same sense as our normal messenger RNA, a plus strand of RNA, it's running in the 5' to 3' direction, it should have no problem getting on our ribosomes, hijacking them, and being able to instruct that ribosome to make those viral proteins. But it's going to be complicated if their RNA is in the negative sense direction, because our ribosomes read mRNA that goes 5' to 3'. So when we have negative stranded RNA as the genome, we got another problem. We're going to see that in order to both make copies of our genome of RNA and also translate it into proteins, we're going to need to express likely two proteins, an RNA-dependent RNA polymerase